In this presentation, we will examine mechanism of action of chymotrypsin as an example of a mechanism of reaction of an enzyme. So chymotrypsin cleaves proteins on C side of aromatic amino acids, aromatic amino acid residues, and those would be tryptophan, phenylalanine, or tyrosine, and also of large hydrophobic amino acids such as leucine and methionine. And it used in its catalysis, it utilizes both acid-base and covalent catalysis. Here you have an example of segment of peptide chain, and you can see that in that chain phenylalanine and methionine are indicated, they are shaded, so that's where chymotrypsin would cleave the chain. It would cleave on the C side, so that means on the right side in each case. Activity of chymotrypsin can be easily monitored by using N-acetyl and phenylalanine p-nitrophenyl ester as a substrate. So this is not a peptide chain. It's not even an amino acid. It's actually an ester, and rather unstable ester of an amino acid of phenylalanine and acetylphenylalanine. So this ester undergoes ready hydrolysis or cleavage upon the action of chymotrypsin. So it's a very good model. And the reason why it's such a good model is that after p-nitrophenyl group leaves, it's in the form of anion, p-nitrophenolate anion. And that anion is colored yellow. It's due to resonance of benzene ring, nitro group, and negatively charged oxygen. So due to electron delocalization. But in the ester form, when this oxygen of p-nitrophenolate is attached by covalent bond to carbon-carbon, compound is colorless. It's only when leaving group, p-nitrophenolate group leaves, that yellow coloration occurs. So from the intensity of the yellow coloration of the solution, we can determine the extent of the reaction. So activity of the enzyme is very easy to monitor. And by monitoring the activity of the enzyme, we have noticed that there are two stages that are evident: a rapid burst phase and then steady state. The mechanism of the reaction involves two steps. Step A is acylation. So acylation forms enzyme-substrate complex, or acyl-enzyme complex. And second step, shown here as B, is deacylation to regenerate free enzyme. So carboxylic acid leaves and free enzyme is regenerated and can participate in the next catalytic step. So these are the two steps listed here. Active site involves a catalytic triad. And that's very common motif in a lot of enzymes, that a catalytic triad is present. Catalytic triad is composed of 3 amino acid side chains. And those side chains are in close physical proximity to each other. Now, they are not close to each other on a chain. So in case, for example, of chymotrypsin, that catalytic triad is formed by side chain of aspartate 102, so 102nd amino acid in a chain, histidine 57, and serine 195. So you see 57th, 102nd, and 195th amino acids on the chain. But chain folds in such a way that these 3 residues are very close to each other. And you can see that they are attached to each other or interact with each other through rather strong hydrogen bonds. Purpose of catalytic triad is to convert side chain of serine, which contains hydroxyl group, into strong nucleophile. So through hydrogen bonding, side chain of serine, OH group, is deprotonated and is in the form of alkoxide anion, which is rather strong nucleophile. And that's what participates in catalysis of the reaction, as you will see in a minute. The mechanism of catalysis is rather complex, and it involves 8 steps. So in the first step, substrate binds, in this case peptide, to the enzyme. It involves recognition of the side chain of the substrate that means amino acid residue of the substrate, with specificity pocket of the enzyme. So in this case, it has to be non-polar side chain to interact with specificity pocket of the enzyme. In the second step, a nucleophilic attack occurs. So nucleophilic attack is attack of the nucleophile, of the serine 195 nucleophile. We already examined that on the previous slide. Catalytic triad makes serine side chain, oxygen of that side chain, negatively charged. It's in the form of the alkoxide. And so as a strong nucleophile, it attacks carbonyl carbon. Reaction is catalyzed or made rather easy by the presence of the oxyanion hole at the catalytic site of the enzyme. We will examine oxyanion hole a little bit later in the next slide. But the role of the oxyanion hole is to stabilize the unstable tetrahedral intermediate. So this is actually how enzyme catalyzes this reaction. But this particular reaction would be rather slow because without the catalyst, formation of this tetrahedral intermediate is actually that high energy part. If you recall from the previous presentation, that point X on the energy diagram where transition state forms, that high energy transition state. Well, here, due to presence of the both catalytic triad that makes oxygen strong nucleophile, alkoxide anion, and oxyanion hole that stabilizes the intermediate, the tetrahedral intermediate, now the enzyme substrate complex has much lower activation energy, and so reaction is much faster. So that would be, this one would be the key step in the catalysis, because this one bypasses that high energy uncatalyzed step that without catalyst would be high energy step. And then in the next step, tetrahedral intermediate collapses. So the collapse of tetrahedral intermediate results in reforming of carbon-oxygen double bond or carbonyl group, and that's accompanied by breaking of the carbon-nitrogen bond. That means peptide chain has been cleaved. And that is assisted by aspartate 102 and histidine 57 of the catalytic triad, because you can see that they now, aspartate forms hydrogen bond to histidine, and histidine in turn forms hydrogen bond to the amine that has been released in the course of reaction. And then in the next fourth step, amine is released. Obviously, amine was held only through hydrogen bond with histidine, and so in the next step, amine is released. In the following step, fifth step, a molecule of water enters the active site. So binding of water simply means that molecule of water enters the active site, and now it binds through hydrogen bond with histidine, the same way as amine before and a side chain of serine used to bind to histidine. So you can see how important histidine is through formation of repeated hydrogen bonds with various reactants that participate in this reaction. By formation of hydrogen bond between molecule of water and histidine, now molecule of water becomes a strong nucleophile. And so in the next step, molecule of water undergoes nucleophilic addition to carbonyl group, and second tetrahedral intermediate is formed. As before, its formation is facilitated by presence of catalytic triad. That means, or actually in this case, only two molecules, two residues, that would be aspartate and histidine, which form hydrogen bond to water, and oxyanion hole that stabilizes tetrahedral intermediate. In the next step, this tetrahedral intermediate collapses, and now molecule of acid is actually released. In this case, it's still there in the active on the active site, but bond to it, physical covalent bond, has been broken. Also note that catalytic triad has been restored. So original hydrogen bonds between aspartate, histidine, and serine residues have now been reformed in this 7th step. And in the final 8th step, acid is released, acid portion of the chain is released, and it leaves the active site of the enzyme, and so now enzyme is effectively regenerated and can participate in the next catalytic step. It can catalyze another catalytic cycle. Here you can see structure of the oxyanion hole. You can see that it is very close to serine 195. That's the one whose side chain is the nucleophile in this reaction. And you can see that NH group of glycine 193, and that's NH group that is part of the peptide bond, and NH group of serine 195 stabilize tetrahedral intermediate through formation of hydrogen bonds with original carbonyl oxygen. Of course, in tetrahedral intermediate, that oxygen is no longer carbonyl oxygen. But through formation of these hydrogen bonds, tetrahedral intermediate is stabilized. And that, of course, lowers the activation energy of the transition state. Now, structure of the specificity pocket is shown here, and specificity pocket is composed of non-polar amino acid residues. It's relatively large, and that means that only substrate that has side chain composed of relatively large non-polar atoms or large non-polar group can nicely fit into this specificity pocket and form numerous hydrophobic intermolecular interactions. And so that's the only one that actually can be held in place by the specificity pocket and allow enzyme to act upon it. And so that's what makes enzyme specific for particular amino acid residues, for cleavage of the peptide bond on the C side of particular amino acid residues. And that means aromatic and those non-polar with larger side chains. This is an example of phenylalanine side chain interacting with specificity pocket. So active site of chymotrypsin contains catalytic triad. This is a schematic representation. I try to be as accurate as possible. So it contains a catalytic triad, and it's shown here in blue. It contains oxyanion hole, shown in red, and specificity pocket, shown in green. And when peptide interacts with the active site, you see what happens. Specificity pocket interacts with side chain of the peptide, while oxyanion hole stabilizes tetrahedral intermediate through interaction with what used to be carbonyl oxygen. And of course, catalytic triad involving histidine participates in the reaction. Histidine is often present in the active site of an enzyme. The reason why histidine is often present is that, in fact, that's the most common amino acid residue that is encountered in catalytic triad. There are numerous other catalytic triads that are present in other enzymes, or just in general in the active site of the enzyme. The reason why histidine is so frequent in the active sites of the enzymes is that it is amphiprotic. Amphiprotic means that it's capable of both donating a proton and accepting a proton. Note that it has NH group, so it could donate a proton, and it has another nitrogen with free electron pair, one that is part of the imidazole ring that has a double bond. So that nitrogen has free electron pair and can accept a proton. In this case, both groups are involved in catalysis. So NH forms hydrogen bond to aspartate carboxylate anion, and nitrogen forms nitrogen of the histidine forms a hydrogen bond to hydroxyl group of serine. So that feature, that property of imidazole ring of histidine fact that it is amphiprotic makes it common feature of the active sites of various enzymes. And finally, we're going to look at some at inhibitors of chymotrypsin, and actually not inhibitors, but just one inhibitor as an example. Chymotrypsin is inhibited by diisopropyl phosphofluoridate, or DIP. It's a transition state analog. Here is its formula. So you can see two isopropyl groups. There is a phosphorus attached with double bond to oxygen and fluorine. Note that this is tetrahedral, that this is stable compound that has tetrahedral geometry. So it mimics transition state that enzyme acts upon. Note that when enzyme was reacting and catalyzing a reaction, the tetrahedral intermediate was unstable and lasted for a very short time. Now, any enzyme has the highest affinity for the transition state. Affinity for the transition state is far higher compared to affinity for substrate or product. And of course, that has to be the case because transition state exists for a very short period of time. And so by having high affinity for transition state, enzyme is very efficient in catalyzing the reaction because it stabilizes transition state, lowers its activation energy, but even with all of that, transition state has very short lifetime and very quickly is converted into product. And enzyme has relatively low affinity for the product, so product leaves. So that way, because transition state has very short lifetime, enzyme is capable of catalyzing large number of reactions. Because a transition state, even though it's very strongly attached to enzyme, exists only for a very short period of time, typically much less than a second, maybe 10 to negative 10, 10 to negative 11th of a second, and collapses very quickly into product. And so product very quickly leaves enzyme. But in this case, we have something that looks like transition state. It's tetrahedral. It has those polar groups, just like transition state. And so it fits very nicely into the active site of the enzyme. You can see phosphorus, oxygen double bond fits very nicely into oxyanion hole. Isopropyl group fits nicely into specificity pocket, but it's stable. And so by definition, since it looks like transition state, enzyme will have very high affinity for it. It will form very strong intermolecular interactions to it. So this one is not going to leave transition state. It's not going to leave the enzyme because enzyme has high affinity for it. So very stable complex forms. And that prevents other molecules from accessing the enzyme. And so this enzyme is effectively deactivated. And so this enzyme cannot operate any longer. And so that's one way to inhibit enzyme, deactivation by transition state analogs. There are other ways, and we'll cover them later in this chapter. So this completes our examination of mechanism of action of enzymes on example of chymotrypsin.